Is no, it won't suffice. But I'm going to try to convince you of some of the benefits of targeting B cells at least. I'd like to start actually by thanking Professor Hammond. We have never met before, but I attribute my move into MS research to you and to your observations of the latitude gradient effect. So you may not know, but I started off my research in the field of UV and UV-induced immune suppression in the context of skin cancers. Working with Professor Halliday and Professor Ross Barnetson at RPA and the University of Sydney. And it occurred to me that there must be some health benefits associated with sunlight exposure in addition to the dangers of sunlight exposure, and that's when I started reading your work. That's where I started moving into the field of MS research, and I'm very grateful for that and your contribution to the field. And thank you also to Simon Hawke for the invitation to come to Orange. I sort of feel like a local. My mother was born and bred in Orange. My wife and her family have a farm in Molong, and we travel to Orange and Molong very frequently. So I love this place, and it's a real pleasure to be able to come and present to this very prestigious audience. So I'm also very acutely aware that I'm the second last speaker. And it's been a fantastic day. So I'm not going to try and show you lots and lots of data sets. I really want to convince you of the need to look at B cells more closely, to highlight the possibilities that there are really two types of B cells that we should be targeting. And the therapies currently have been almost solely targeting the pathogenic or the bad B cells, as I call them. But we really do need to start turning our attention to understanding those other types of B cells which actually might be affording some protection from multiple sclerosis. And my student Felix has already sort of started that off very nicely by highlighting our observations in the context of UV and how UV activates a unique type of regulatory B cell which in our mouse models can be transferred and can transfer the protection from a CNS targeted autoimmune disease. And we are now moving these studies into humans working with Simon and George, but others looking at UV irradiated patients. It was alluded to earlier in today's session of the Prevance trial, the vitamin D supplementation trial in CIS. And we are also participating with Robin Lucas and Prue Hart in investigating PHOSIS, which is phototherapy for CIS, looking at whether phototherapy, giving UVB to CIS patients, can prevent the onset of multiple sclerosis. I'm not going to talk to you about that today. What I do want to talk to you about is just to remind you about some of the stunning success stories when it comes to drugs which target B cells, and gave us the real first evidence that B cells were important in this disease, because prior to this, MS was almost entirely considered a T-cell driven disease. So all of you in this audience are all very familiar with the early work on rituximab showing the spectacular success of that drug in MS patients, and of course the more recent evidence in progressive MS particularly of the next generation of drugs which target CD20 on the surface of B cells. And we've seen this figure already today, but I do think it's really quite remarkable to keep this in context, and I think speakers throughout the day have highlighted the need to do that. And I think this graph really highlights it very nicely as to how targeting B cells can be very effective. And what this evidence really has spurred some people on to doing is targeting B cells and the very growth factors that they need for their survival, and get rid of all B cells. And the other reason why this is an important endeavour is that almost all the successful drugs that work in MS, at least at some level, will target some aspect of B cells, whether it's their trafficking, whether it's their activation status, or whether it's their depletion. And so what has been proposed is to target the cytokines that B cells need for their survival. And in so doing, you'll get rid of not just the CD20 positive B cells, but all B cells. And some of the most important cytokines that B cells need for their survival include BAFF, or B cell activating factor, and APRIL. These are crucial cytokines for B cell survival, and some have developed antibodies that target these cytokines, and these have been proven successful in some diseases like SLE. And others are using what is known as TACI, a fusion receptor, or tacicept, a decoy receptor that blocks all signals through those receptors into B cells. Now, this audience probably already knows that this approach doesn't work. And in fact, the dangers of depleting all B cells is highlighted really by this particular failed study. And it gives us a real hint and a clue as to where some of the good B cells might be. And in fact, we do have good B cells and bad B cells. And targeting all of them is clearly not a good idea. And targeting one is good, but it's not the best that we can do. And so, as you've already heard from Felix, we're making great strides using the latest cutting-edge technologies to interrogate the B cell populations to work out which ones are the good, which ones are the bad, and how can we optimise this for therapy. So, I would like to take you all back on a bit of a journey. Now, if any of you have these shirts in your closet still, looking at the gentleman in the audience, your wives have probably already encouraged you to get rid of them, or if they haven't already, you probably should tell her that you still have them. But where I am taking you is back to the 70s, and there'll be some good reasons for that in just a few moments. But let's just explore the 70s in just a little bit more detail, because that was the decade that I was born, and it was alive with colour. And it was a time for pipes and slippers, apparently, and very garish ties. And it was really very easy to live in your one-piece suits. The onesie is coming back, and I think that's the one thing from the 70s which we should not be restoring, the one-piece, the one easy piece. And of course the matchless suit, which thankfully never came back, and that's quite a horrible. And of course pirates were in for some reason. This guy, I think, is saying, take my eye out now, I don't want to be seen in this particular get-up. The reason I'm taking you back to the 70s is to remind you of some of the research and discoveries that were going on at that time, particularly in the context of immunology and the discovery of regulatory cells. And I think it's important for us to realise that we're not reinventing anything here, that we've known about these cells for a long time. What we did actually was forget them for a long time. And I'll come to the reasons for that in just a few moments. So this is really one of the first papers published in immunology, a really seminal piece of work showing that tolerance can be induced by cells that were derived from the thymus. And over the time, as the 70s went on, in the Journal of Immunology, they started to call them suppressor T cells. You notice that they were called suppressor T cells at that time. A remarketing exercise occurred in the late 90s to call them regulatory T cells, but at the time they were called suppressor T cells. What you may not know is that at around the time that suppressor T cells were discovered, suppressor B cells were also discovered. This was the first evidence that I could find, at least in the mid-70s, showing that there is a suppressor cell that exists with Ig-type molecules on its surface. Others, a few years later, Katz, Parker and Turk in their seminal Nature paper really were responsible for highlighting the important role that B cells can play in modulating or regulating immune responses. We have been unable to implicate the antibody, though, in the mechanism of this suppression. So that's a really important observation, because what it shows us is, one, that B cells are immune regulatory, immune modulatory, and two, they do it via a mechanism which they didn't yet understand, and we still don't understand the mechanisms by which regulatory B cells work. So the real question is, why were suppressor cells abandoned in the 70s and 80s? Well, this paper really hypothesises there were four main reasons, and really important reasons why they were abandoned, and these reasons also highlight why we return to them. So there was a failure to find reliable markers that could distinguish suppressor T cells from other T cells. I would argue we're at that same point with B cells. There is an absence of reliable markers that allow us to distinguish regulatory B cells from other B cells, but we're making inroads to discovering what some of those markers might be. There was ambiguity in the molecular basis of the suppression. We have ambiguity for regulatory B cells right now as to the mechanism by which they suppress as well. There was great difficulty, a technological difficulty, in preparing antigen-specific suppressor T cell clones that would allow them to fine-tune their analyses. So being able to single-cell sort, apparently, was required to be able to definitively show that these cells were regulatory. And finally, and perhaps most pertinent for this audience, was the failure in the clinic to actually obtain definitive evidence for suppressor T cells as a primary cause of any immunological disease. Now, we know that that's clearly not the case anymore, because in the mid-90s, almost 20 years later, it was found that yes, we can clone T cells that are regulatory, and they're responsible for mediating suppression from important diseases. T-cell, antigen-specific T-cell subsets in the context of important human diseases. So we're talking mid to late 90s now. But again, what you may not realise is that around the time that we were rediscovering regulatory T cells, rediscovering and rebranding regulatory T cells, B cells were also being discovered that could exert different effector functions via the production of cytokines, not antibodies. And so it was quite clear that B cells can exert dual effects on cell-mediated immune responses. they can promote adaptive immune responses. That's what they're most well known for. And they do that in a number of different ways. And they can also suppress adaptive immune responses. They can present antigens to T cells, and they can be in the CNS or outside the CNS. They can, of course, produce antibodies and cytokines that promote Th1 and Th17-driven immune responses. How they suppress immune responses is not fully understood, although it's quite clear that they do it via the production of some cytokines, and which cytokines they produce is not entirely clear. IL-10, IL-35 and TGF-beta seem to be particularly important cytokines in this context. And they also appear to be collaborating with regulatory T cells to modulate their function, and so it's clearly a crosstalk and collaboration that's occurring rather than just all being a B cell-driven phenomenon. So at around the time that these B cells were being rediscovered, some really important work was being done by Charlie Janeway's group. Some of you are probably familiar with, may have even known Charlie Janeway. I know Charlie Janeway from his, of course, his textbook, Immunobiology, Janeway's Immunobiology. So his group was one of the first to discover a role for B cells in modulating a CNS-driven autoimmune attack. In this case, it was the experimental autoimmune encephalomyelitis model. I just want to quickly show you just one figure from that important paper. So this is the clinical score of EAE on the y-axis and days post-induction. So for those who are not familiar with EAE, it is an induced disease. And they induced the disease in two types of mice, wild-type control or a mice which is deficient in B cells. They don't have any B cells. And the first thing to point out about this particular experiment is that you don't need B cells in order to get EAE to take hold. So B cells aren't necessarily responsible for initiating the disease. What you do need B cells for, as you'll see here, if you don't have any B cells, you never resolve the disease. In normal mice, the disease is resolved after about 3 or 4 weeks. But in B cell deficient mice, you don't get resolution of the disease. And so what this showed us was that B cells were actually required to place limits on the severity of the disease. More recently, Thomas Tedder has been pioneering the work in this area in mouse and in human studies, identifying important roles for regulatory B cells in the context of this type of autoimmunity. And I want to very briefly take you through some of the important findings that they have shown in the context of the kinetics of when B cell depletion occurs and the implications that that has on the course of the disease. So in this first experiment, the arrows indicate when a B cell depleting antibody was injected, so mouse rituximab, if you will. It was depleted before the disease was induced. And what you can clearly see is if you do that, here in black are the mice which received the B cell depleting antibody, the mice got more severe disease. And so B cells were actually required to place limits on how severe that disease was going to be. So this is very reminiscent of Charlie Janeway's first observations. If you deplete after you've induced, but before the disease manifests, it had no effect on the disease course. If, however, you inject the B cell depleting antibody once the clinical score starts to manifest, you can actually protect the mice. This is what we see in MS patients. If you deplete the B cell depleting antibody once the disease starts to resolve, again, it has no effect on the disease course. So this is an important observation, albeit in mice models, showing that B cell depletion has different effects depending on when you decide to deplete the B cells themselves. Others have shown that the production of IL-10 is really important for this particular observation. And in this particular study, what you can see here is if you transfer B cells that can produce IL-10, you get resolution. But if you transfer B cells that can't produce IL-10, you don't get any resolution. And so what it clearly shows is that IL-10 appears to be an important cytokine that B cells need to be able to produce in order to protect the mice. Others have taken this even one step further and can induce these types of regulatory B cells in vitro cultures and get them to expand to massive numbers, transfer those B cells via adoptive therapy into mice and can transfer the protection. I'm going to show you just 2 studies which are doing this because I think this is actually one of the areas in which it could be beneficial for MS patients if we can understand which B cells are protective. So in this particular way of inducing B-regs, they call them gift B-regs, you can see here that every time they injected the B cells, the B-regs, the disease was suppressed. So here we have the control mice up here, which go on to develop a relapse-remitting style of disease. And here are the mice which were fortunate enough to get the B cells. And every time the disease starts to come back, they hit them again with another dose of the B-regs, and ultimately you can cure the mice of this particular type of disease. Others, including Thomas Tedder, who I've already mentioned before, are using a slightly different way of expanding the B-regs, using IL-21 in particular. And this just shows you the massive amplification in the numbers that they can generate. Notice that this is a log scale. So this really is a massive expansion of B-regs. So this is an important proof of principle showing that we can generate these B-regs in culture to levels that are going to be required to inject into patients. I'll skip over that one. So it's quite clear, therefore, that B cells exert dual effects on the immune response, and we have MS-promoting B cells. They are clearly going to be important cellular targets for the treatment of MS. From the evidence that we have in the literature, and I'm going to return to this at the end, and hopefully we can have a discussion about this, that they do not appear to be reliant on BAFF or APRIL, but they obviously must express high levels of CD20 in order to be susceptible to treatment from rituximab and ocrelizumab. And so depleting these B cells is going to be very, very important and to continue to be an important therapy. However, we also have MS-protecting B-regs, and this really is now an open field, a black box of discovery. And the way that they suppress is completely unknown. And they are going to be equally important cellular targets for preventing MS. From the clinical studies, it would appear that B-regs might be more reliant on these cytokines, BAFF and APRIL. The etacicept trial tells us that. And they may express very low levels of CD20, or they may even be negative for CD20. Thus, they are not depleted by rituximab or ocrelizumab. So, in humans, the most important question is, well, where are B-regs? In which of the different types of B cells that exist and are known, where do the B-regs lie? The short answer is everywhere. We find them in every compartment. So this is the pre-B cell, just after it leaves the bone marrow, and it undergoes a variety of different maturation stages depending on its location, whether it's in the spleen or the lymph node or the blood, or whether it engages with a T cell and forms a germinal centre, gets signals to become a memory cell, and whether it actually induces long-lived antibody responses. So some people have, in different contexts, not necessarily in MS, but in other autoimmune disease contexts, in the context of cancers as well, some have shown that B-regs exist within the immature population of B cells. Others, and we would concur with this in the context of MS at least, and Felix's work at this point, would suggest that some of the B-regs lie in the transitional area of the B cell lineage. Some, in different contexts again, have shown that actually no, the B-regs lie in the memory cell compartment. So this makes it, as an immunologist, this makes it really interesting, but as someone who's trying to find a B-reg that helps MS patients, this is extremely frustrating because it's not yet clear which are the good B-regs in the context of this disease. And so differentiating the good from the bad is clearly very challenging, because if you just look at a B cell down a microscope or down a flow cytometer, they all look very, very similar. The trick is actually using the cutting-edge technology that we're doing to actually identify which one are the B-regs. They are not necessarily going to be there all the time, and they are going to be quite difficult to find out, to look at, to interrogate, and to work out how they actually perform their functions. So this leads me to my final slides, where I want to sort of talk about some of the different ways in which people are thinking about B-regs right now. And there are three models that have been proposed by primarily Claudia Maury's group in the UK. And I've borrowed her slides to sort of promote some discussion about where we think some of the B-regs that are important in MS might lie. So the first model that she's proposed is that different B-reg subsets arise from individual progenitors. So you have a pre-B-reg that leaves the bone marrow and is destined to become an immature B-reg or a B1 B-reg or a B10 B-reg. And that there must be a transcription factor or some gene or some protein that we could find inside the pre-B-reg that would allow us to actually zero in on that particular subset and promote that subset or target that subset in some way. Unfortunately, there doesn't appear to be any evidence for this model. Model 2 is that all the different B-reg subsets arise from a single progenitor and express a single transcription factor, very much like the T-regs with their FOXP3. And that given, depending on what signals they get, they'll become all the different types of B-regs that we know and love. And that the B-regs are not necessarily terminally differentiated, they can exhibit suppressive functions at different stages of differentiation. So there is some evidence for this, at least in the different B-reg populations, whether they be immature, naive or memory, but there is no evidence for a universal transcription factor which allows us to identify them, which makes our job much, much harder. The third model, which of course is the hardest to interrogate, but for which we actually have some evidence, is model 3, that any B cell can become regulatory upon exposure to a specific environmental stimuli and can exhibit suppressive capacities. These B-regs are likely to be short-lived. In other words, once the inflammation goes, so too does the B-reg. They're no longer needed, they're no longer sticking around. And in the context of an inflammatory disease like MS, there may be an absence of one of these cells, and we're really working very hard to try and identify which one of these we need in the context of MS. So this is my final slide, and this really sort of sums it all up in terms of where we think we need to be looking. And we're going to be guided a lot by the clinical evidence. So we know that many of these different types of B cells, depending where they are in their lineage, whether they've just exited the bone marrow, whether they're returning to the bone marrow to become long-lived plasma cells, we know that many of them express high levels of CD19 and CD20 at this stage. And so this is probably where the vast majority of the, I'm going to call them bad B cells, probably lie in the context of MS. Now, I'm very reticent to use the word bad and good because, it depends on the disease context. So in the context of cancer, these are probably good B cells, probably helping to destroy the cancers. But in the context of autoimmunity, these are likely to be where the bad B cells lie. Alemtuzumab, of course, depletes almost all of them, except for those at the very highest level here of differentiation in the plasmablast and plasma cell range. Nearly all of them are blocked in trafficking by natalizumab, but it's down here, the reliance of some of these cells on signals through BAFF receptors, signalling of BAFF and APRIL in particular, evidence from the etacicept trial, tells us that the potential for this end of the spectrum to house the good MS protecting B-regs is where we are particularly focusing our attention with regards to identifying where some of these cells lie. And with that, I'd like to thank you for your attention.
Thank you for an excellent talk. Are there any questions?
Apparently not.
Can I ask one, Scott?
Yeah.
I mean, do you think that 10 years' time there'll be much more tailored therapy for these different subsets to promote and deplete?
I think tailored, yes, but tailored at both ends. More targeted therapy for those that are actually causing the problems in MS, and attempts to actually promote the good B-regs. I think the reason why we're seeing some of the spectacular successes with the B-cell targeting therapies is that by depleting those B cells, you're actually creating a bit more space for the B-regs to exert their effective functions, but also you could potentially be selecting for some of those B-regs as well. So I think we need to focus more attentions on how do we actually tickle the B-regs a little bit to get them to go. Yeah. I'm actually going to let Simon answer that one if that's okay, because my understanding is we don't have any ocrelizumab patients at the moment. Is that right, or we do?
It's in the process.
Yeah.
Yeah. So there are some. I think 150 people have been treated so far in Australia.
Yeah.
So it's just being.
It's the early days. The short answer is yes, we would like to, absolutely, but at the moment our efforts are focused on probably alemtuzumab and rituximab and fingolimod. Yeah.
So with alemtuzumab, Tregs come sort of depleted and B cells come back early.
Yes.
There's increased risk of autoimmune. Do the B-regs not come back?
Well, actually, Felix will hopefully be able to tell us very soon when he finishes his analyses. But if you remember from Felix's slide, in one of the alemtuzumab group, there were B cells. So we saw the same thing that Alistair Cole sees with the B cells coming back early. What we don't yet know is whether within that B cell pool that comes back early, whether the B-regs are in there. But we hope that Felix will be able to tell us sooner rather than later. Yeah.
Great. Thank you very much.
I do remember when I started my PhD, Simon was still finishing his. So I've known him for a long time, and I didn't It was a great shame when he left Sydney. What I wanted to talk about is predominantly about T regulatory cells, but I just thought I'd go over a few of the issues that I think that some of them have all been covered extremely well in the talk beforehand, all the talks beforehand, but just how they interact and what I think could be important. So, first of all, what do actually T cells... So some of the things I just wanted to do was some of the immunological facts that sort of underpin all of the observations that we might make from disease course, therapy, genetic studies. There's a lot of circumstantial evidence that T cells are really very critical, and these include the genetic association studies, the pathology studies, the animal models, the effects of therapy on the disease which target some aspects of T cell function, the observations that high parasite loads protect against MS, and I'll just go into how that might happen, and then some of our peripheral blood studies, which include studies on effector and regulatory T cells. B and T cells have many interactions. It's worth remembering that B cells are the most potent antigen-presenting cell to a T cell that exists in the body. So they only become really activated if they're exposed to T cells, and either to expand or to become plasma cells. So that they're a very integrated sort of couple of cells. They've got really highly specific receptors, which are the B receptor, the antibody, that can bind to antigen anywhere that they encounter it, and then they can process it and they can represent it on their MHC class 2. And B cells that are infected with Epstein-Barr expand a great deal. Michael Pender tells me that in a normal population, so most of the people who don't have MS or don't have lupus, they've really only got about 1 or 2% of their B cells with EB virus inside them. But an MS patient has up to 10% circulating. So I think, and those cells are possibly turning over and are in a state of slightly increased activation. Now, how is all this sort of regulated? T regulatory cells, we believe, are antigen specific, can control both B and T cell responses really well. Antigen specific T regulatory cells can suppress a ratio of 1 to 1,000, and they can also travel to the specific site that the inflammation is occurring in. If they're deficient or underactive, both T effector and B T cells will expand. Only about 5% of circulating CD4 cells in any body are Tregs, and this ratio is kept extraordinarily stable. Just by way of history, so people understand where we've come to with this antigen-specific Tregs. They were first actually described in Sydney. First of all, CD4 cells were identified as being antigens containing the antigen-specific suppressor cell, and that was by Bruce Hall in 1985. And in 1990, he described that these cells had CD25 on them, and they were antigen-specific, and they could suppress a fully allogeneic transplant. The naive cells, the naive non-specific cells that Sakaguchi described, were described in 1995. If we think about the power of the immune system, if you have no T cells, people die. If you have no regulatory cells, such as occurs in the IPEX phenomenon, patients die. If T cells are overstimulated, and there was a really unfortunate human experiment with this, where patients were given anti-CD28 that turned on nearly every T cell in the body. Within 10 minutes, the patients were desperately sick, and within 28 hours, some of them had died of multi-organ failure. So our immune systems are constantly, totally being regulated. Generally, some of the systems that you're used to regulate it are pretty simple. It's just, one of them is that effector cells, both B and T cells, die when they've eliminated the antigen. So you get a cold in your nose, you kill it, and those effector cells go. They generate memory cells which stick around for a long time, very useful for measles and tetanus. If antigen isn't eliminated, that's when we start to have the expansion of antigen-specific T regulatory cells to stop major immune injury. And the most powerful over a prolonged period of time here are the T regulatory cells. The thymus produces two lineages of CD4 cells. They're either Tregs or they're not Tregs. And they have these little markers on them, FOXP3, 127. I'm not really going to talk about the effector lineage cells that can convert to an induced regulatory cell. They're much less stable. They're very important, but they're less stable, and they're probably not really critically involved in long-term sustained inflammation. I only put this slide up to show some of the names of the markers that control this interaction, I also put this up for a similar reason. But one of the things is this is the pattern of behaviour, where a pattern of how we amplify just effector responses from a T cell. So we have this ordinary T helper cell. If we use IL-6 and IL-1 and antigen, they'll develop Th17 cells. Antigen and IL-2 will have Th1-like cells, and antigen and IL-4 will have Th2-like cells. Now, actually, if you look at T regulatory cells, exactly the same type of thing occurs. So we've published this from 2009, was the first time we described the first component of this, and some other people have found some of these sorts of cells in various situations. But basically, and it applies in transplantation models and in autoimmune models. Basically, if you take the NT-reg, the naive T-reg, out of the thymus, and you expand it with antigen, if you expand it with IL-2 and anti-CD3, you'll expand lots of these sorts of cells. They'll be polyclonally expanded. They suppress it 1-to-1. They don't cross into tissues. If you expand them with antigen and IL-2, after 4 days, they have some other receptors on them. They have the gamma interferon receptor, and they might have the IL-12 receptor. If you expand them with antigen and IL-4, they get the IL-5 receptor on them. This is the only T cell in the body that has the IL-5 receptor on it. It's present in rats and it's present in man. If you go back and you take these cells and you just add the antigen and gamma interferon, or the antigen and IL-12, you can produce a Th1-like Treg here, and these are the cells that start to suppress at 1 to 1,000. And actually, we have shown that if you take these cells and you add IL-5 and antigen, you can produce a Th2-like Treg cell. Now, from a facts point of view, it's not possible for some of these receptors, they're not in high enough things to be easily identified. But what people have started to do is use the chemokines that are on Th1-like effector cells and to see if you can track where they're going. So they've got CXCR3 or CCR, particularly the Th17 one is thought to have CCR6. And we're still looking for a reliable chemokine in the human to track these type of cells, because the CCR8 that's reported doesn't seem to work. If you think of all of those cytokines and receptors, nearly all of them have been found to be associated with multiple sclerosis, really suggesting that that immune circuit is in some ways critically important. Now, we've been studying EAE for quite a long time, and some of the things we've shown is that animals that have recovered from EAE have antigen-specific Tregs in them. you can take them out of the recovered animals and you can put them into the new animal. You need quite a lot. You have to wait for quite a few weeks after the animal's recovered, but they're there in enough numbers to actually put them in. If you just take the same number of naive Tregs out, it doesn't work. But of course, we can't in any way use recovered humans to treat other humans. So we thought we'd see if we could do this in vitro. So using the model from before, we've grown Treg cells either with antigen and either IL-2 or antigen and IL-4, and then given them to animals to see if we suppress the disease. If the Tregs are grown with antigen and IL-4, which is one of the experiments we've done, you need very few numbers to do that. So this is our first experiment where we've given cells that have been given IL-4 and antigen and are given as soon as the animal gets sick, and we substantially ameliorate the disease. You can block this effect if you block if you don't grow it with the right antigen, this effect doesn't occur. So we then went on, that's sort of really giving this type of cell what we've called a TS2 cell, and then we've gone on and made some of these. So you go from 4 days for this cell and 3 days with antigen and IL-5. And when you do that cell, you only need 1 million cells into a rat. A rat has got 300 million CD4 cells. A significant number of them are effector, and you can substantially reduce the disease. We thought maybe we'd just use the cytokine and see if we could expand that cytokine, use that cytokine to expand the cells that are already there. And if you give IL-5, you actually just prevent the disease occurring. We've done it in experimental allergic neuritis, which we published in, but it also applies in EAE as well. So we also thought, well, what are the other conditions where IL-5 is around a great deal? And one of them is in parasite infestations. So MS patients with eosinophilia, this was referred to by Coriol's work, which one of the speakers beforehand described really well, Mac, where he showed really clearly that the MS patients that actually had the eosinophilia had substantially reduced amounts of MS. And that he also went on in another paper and had treated some of these patients and their disease came back. So parasite infestations induce Th2, which make all these cytokines. The eosinophilia is induced by IL-5. And we have found that antigen-specific Treg cells induced by IL-4 show that they have IL-5 receptor on them. So we thought that maybe we could look in what happens in the parasite animals. We could look at what happens if we give parasites to animals with EAE. This is just another study looking at the prevalence of MS per 100,000 of population, and you compare it to people if they've got a parasite. And in fact, if you have if there's a country with high parasites, you virtually don't have MS. So some of the things I think is happening as we westernise countries is that we clean up the water supplies and we improve washing and clothing, and people's parasite load drastically drops. Of course, there's many other things that occur and affect the biome as well, but I think that might be one of the things. So we've done a lot of experiments now looking at the parasite effect on EAE. And this parasite is not, it dies quite, we had to use a parasite that's not terribly dangerous. If we give them the parasite, the animals don't get as sick. If we block the IL-5, these animals retain the same amount of sickness. I haven't got the other controls in this talk, but we've gone ahead and blocked it. The effect is blocked if we don't give, if animals have no Treg cells. It's also blocked if they don't have IL-4 in this situation, so can't develop the first component of that Th2 type Treg. So we thought that overall it showed that cytokines from Th2 cells can produce antigen-specific Tregs. So I just wanted to go back and say, well, how can we apply any of that to the human population? So what we thought is, could we try and find what is happening to the Th-like Tregs in MS patients using various markers that might actually identify activated Tregs, not just the naive Treg population. And generally speaking, most of the studies in MS have looked at the naive Treg populations, which are essentially the same in most studies, although they're less suppressive. So I'll just keep going through this. So we used CXCR3 to identify Th1 and CCR6 to identify Th17, and we used CD45 to look at the various levels of activation. And as the T cell becomes more activated, it loses more and more of that huge CD45 receptor on the outside. So what this is some of the things that we did. First of all, we just had lymphocytes. This is a fax thing. We stained them, separated them for CD25. Then we used FOXP3, and then we used of that. You can see once you've got this population and you run it against CD45, you end up with 3 different populations. And generally, the previous studies have just looked at this population, but there's a population what we've called 3 and a population 2. And then if you look at that, there's some of them are CCR6 and some of them aren't. And so this is what we've... what we aim to do is just compare the Treg frequency and phenotypes in healthy donors and MS patients with a focus on seeing if we could assess the Th1-like Treg. And we also thought, well, because alemtuzumab is suggested to substantially alter that, to look at over a longitudinal period of time what happens with alemtuzumab. And we thought there would be a difference. So far, we've managed to study 31 MS patients who are switching therapy. Most of these have had some previous therapy, but included in them are some patients who've never had any therapy at all. And the results of these patients, of the never had any therapy or the others, are very similar. We've got so far 4 MS patients. We're aiming to have 10, and we've got healthy controls and we're getting more. So we plan to do all of these things, which we did. And there were 3 populations of interest just to go over. So we're going to look at population 1, population 2, and population 3. So first of all, if you look at healthy donors and MS patients, there's a really substantial difference, particularly in population 2 and 3, but not really in population 1. So this happens to be the more activated component of the Treg population. There's more of them. So that is significantly expanded. And if you look at the CCR6 component of that population, there's maybe a little bit less in the population 2 of the Treg cells that would be deviated towards against Th17 type cells. Now if we go on and look at pre and post alemtuzumab, this is 0 and 3 months. This is what's happened overall at 0 and 3 months. So there is a significant increase as a percentage. But interestingly, the percentages there's really no change in population 1, the naive population. There's quite a marked increase in population 2, and I'm unclear about what's happening here. If you just look at CCR6, that's also increased, whereas previously in the ordinary MS patient it tended to be lower. And CCR7 drops drastically. So these are the recirculating cells that fingolimod tends to affect CCR7 populations. I actually thought that one of the things that we should do is look at the ratio using the absolute numbers use the ratio of population to the effector cells, because it's a ratio probably effect that is important. And there is a significant increase in population 2 in all of the patients bar 1. And quite interestingly, actually, this patient really did not respond to alemtuzumab. So I don't know whether that really means anything. There's not that many patients that don't respond, but there are a number. And I think if it turned out to be true, it might be useful rather than patients just having multiple courses of alemtuzumab, which increase their risk of side effects. So, first of all, overall, population 2 does increase. It may just be a reactive response, which is what I think it is. There may be a genuine deficiency of the CCR6 component of that population 2. And so there may be a deficiency of Th1-like Tregs. After alemtuzumab treatment, there's an increased percentage of ordinary Tregs, that's consistent with other studies, and there's an increased expression of CCR6 expressing Tregs. I think I'll just go through that. We just wish to keep on with, we need to obviously get some more numbers because this is a preliminary study. I should just, this is from ECTRIMS this year with David Hafler presenting some of his preliminary data, and I was somewhat reassured to know that he had very few numbers as well. He's just looking at the myelin reactive T cells, not the regulatory cells, but he actually showed that the effector cells that carried the tetramers are co-expressing CXCR3 and CCR6. That's here, the co-expressing ones. And we found some of our T regulatory cells co-expressed. And he also found that there's a very significant difference in the subsets between MS patients and non-MS healthy controls. So when you start looking at the subsets of using some of these fine markers, you find really marked differences in MS patients to healthy controls, suggesting very significant immune reaction in the periphery of the patients. So first, just in summary, I'd like to say that the regulation of the immune system is very difficult to study because many of the effector mechanisms that are also utilised by the regulatory cells as well, almost identically with a few, so that any immune action you take against it, so you may actually interfere with regulatory cell function as well. Certainly, I think one of the interesting advantages of giving an anti-B cell therapy is that you are not interfering with that T regulatory cycle. Another useful point is to just bear in mind that activated regulatory cells die even inside if there's no antigen and cytokines, so that you can't just take them out and look at them. And overall, I thought that if you remove the antigen presenting cell, and if you could remove it perfectly, that would be excellent and a treatment, and that if the only antigen presenting cell was the B cell and we removed it perfectly, that would be sufficient. But if we can't, which I think will be impossible, the stuff that remains will go on stimulating T cell responses and will require T regulation to be controlled. In summary, these are some of the other people that have helped do all this work, including 2 medical students, Andrew Lamb and Christopher Chu. Thank you very much.
of the controls was quite substantial. Would you like to comment? I'm assuming they're age-sex matched.
No, I think that this is this is a preliminary study. If all of the, just mostly all of the, that's what we've found so far statistically. It may not be correct, but the major difference appears to be in population too. Generally speaking, people have found that there's no difference in number of T regulatory cells between normals and MS patients, but that they usually, if tested in a lot of in vitro mechanisms, they don't suppress as well. One of the things I would say about that is that if you're trying to test suppressor capacity of antigen-specific cells, they don't behave in the same way as naive T regulatory cells. So I think the test might be wrong. I think they may actually suppress quite well, but we haven't been doing the right test. As with the B regulatory thing, there's a lot of issues with this, and finding the right surface markers to identify them has been quite a struggle.
Has there been any reports about CCR6 positive Tregs being deficient in MS patients?
No, there hasn't. So I think that the information that David Happler presented, where he's showing that the effector mechanism, the effector T cells that he believes are important are burying both chemokine CXCR3 and CCR6. That's new data, and when we reviewed all of the literature about T regulatory cells, nobody has looked at the chemokine receptors on T regulatory cells.
Just another quick one. For the experiments that you show about helminths and IL-5, and this is suppressing the inflammation, do you think this is because of the production of Th2-like Tregs, or this is because of shifting the balance between Th1 and Th2? Well,
we believe it's because of your expanding antigen-specific Th2-like Tregs. You've got to bear in mind that this entire process would actually lead to polarization, because the more Treg you have of Th2, the more you will deviate the effector mechanism as well. So I didn't include, that's just come out in Frontiers in Immunology this year, and there are all the controls in there that would assist you, which I didn't include.
If you, just make a comment, if you deplete CD25 cells, parasites do nothing.
Yeah. Yes. So in that model, take the Tregs out.
Then this is not because of the balance.
And then if you take out the Tregs, the CD25 cells, they're like Th2 Tregs. They express the IL-5 receptor. They respond to specific antigen if IL-5 said. So there's an increase in the number of antigen-specific Tregs that are responsive to IL-5 and the antigen.
How dependent are your antigen-specific Tregs on the presence of antigen?
100%.
So if you don't have any antigen presentation, you take antigen away, they die?
No. So if you go to that sort of the cartoon I had, so we've done all of those experiments multiple times. So if you have just IL-2 in there, all that immediately happens is you've just gone back to, you just expand your naive Tregs. So you've got to consider that actually that population, that 5% of Tregs, is got a whole bunch of cells in it that are naive, some are antigen specific. The other thing that's important though is you've got to have the antigen in there. But after you've got to that first phase, if you keep the IL-2 in the second phase, you lose it again. You will not get a Th1-like Treg. You've got to take the IL-2 out. And the same applies with the Th2 phase. The IL-4 has to be taken out. And in fact, if we think about the body, IL-2 and IL-4 in particular, they're quite short, in an immune response, they're quite short-lived cytokines. They don't hang around for days and days. They don't hang around for weeks. So this is why There's reasons why they don't, but it's a phased response, just like with effectors. So, and I think that that's one of the problems where people can't generate these very, very high potency T regulatory cells.
That's what I was getting at. I understand the antigen dependency to induce them and for them to have their function. But once they've been induced, if you remove the antigen completely, do they die?
They do. They die. They die. I mean, probably the best example, or sorry, one of the original examples is the original 1990 paper. That's Nick Pearson Hall, and you generate these cells. You put them into an animal that's got a graft. The graft survives. If you take them out for 3 days, they'll die. If you keep them alive with Conné soup and antigen, they'll live. Well, at that point, we didn't know what that cytokines were, but it wasn't IL-2. And it's a whole bunch of things that's led us to that. So the antigen's really important. And I should say in my animal studies, I haven't used a micropeptide. I haven't used 1 to 11. I've used myelin basic protein. I think, like, one of the problems I think is that in a whole lot of different humans, there'll be multiple different organ-specific antigens because of epitope spreading or HLA types or presentation, all sorts of things. So I think that we could make in vitro T regulatory cells, but to brain or to kidney or to whatever.
Sorry?
I mean, taking on that point, I mean, presumably with the parasites, you're inducing antigen-specific T regs to the parasite.
So they do that.
No, we do that as well.
But I mean, in the natural situation in the human, but then are they going to get into the deep cervical lymph nodes to?
No. So if I can just so you've got a parasite. You do make antigen-specific Tregs, but you've got to remember you make IL-5 because if you have a big parasite infestation, you've got eosinophilia, you've got a lot of IL-5. And you should remember in those countries they don't have a lot of asthma. They don't have a lot of any autoimmune disease. But I think what happens is that if you have any autoimmune response, you've got a cytokine that is supporting the expansion of Th2-like Tregs for whichever antigen it is.
Simon, in the animals, there's 2 responses. There's one to the parasite, there's one to MVP, and there are Tregs to both of them. So in parallel, the antigen-specific one to the parasite's made, but the antigen-specific one to MVP is a separate process. So there's a spillover of IL-5, you just make more of the MVP bystander effect.
Yeah. Is that clear? Maybe.
There's a picture
Hope so.
in the paper that explains.
Yeah.
Con?
So with alemtuzumab, that it's clinically failed, saying that the Tregs do not increase.
In population 2, they didn't go up, no. And not much in anything, actually.
These are patients where they got a normal response in terms of immunosuppression, but then 3 months later, 4 months later.
Yeah, she had standard obliteration of of all her cells. There was no observable difference. It's just her, from what I could find, it's just her regulatory cells in that population did not expand. I have no idea why it didn't. Yeah.
And she didn't do well clinically.
No, she didn't do
That's
well.
really important because
Yeah.
you're only a handful of
Well,
patients.
it's only one patient at this point. Like, I need another one. I should say the other thing about we're using chemokines. Chemokine receptors on the surface of these cells are said to be very, very. These are big activated cells. They're said to be very brittle and that they get lost if they're frozen. So we're handling all of this fresh. We are doing some frozen experiments. If it works on frozen cells, it'll be a hell of a lot easier. But at this point, it might have to be done fresh. And most of the samples of nearly every clinical trial Treg study has been done on frozen samples.
Okay, well, I think we should thank
Yes.
the speakers very much for an extraordinary session.